Hi everyone, today we're going to be performing the Mystery Carbohydrate Lab. This is a lab that uses a solution called Benedict solution and an iodine solution to test various everyday carbohydrates to determine what types of sugars they contain. So if you've ever wondered what sort of sugar specifically, whether it's monosaccharide, disaccharide, polysaccharide, is found in things like honey, molasses, or maple syrup, this lab will show you how to do that. So we're really encouraging scientific thinking here. A quick explanation of the Benedict's test. So Benedict's solution is blue in color, and to perform this test we will mix a certain amount of Benedict solution with a certain amount of mystery carbohydrate solution, and after mixing those we'll boil them for 5 minutes to get our result. Okay, so this is what a positive test and a Benedict's test will look like. You'll mix the blue Benedict solution with your mystery carbohydrate solution, and if it is a monosaccharide, it'll test positive in the Benedict's test, meaning it'll turn orange after 5 minutes of boiling. So the Benedict's test will tell you whether or not a solution contains monosaccharides. It'll also tell you if the solution contains some disaccharides. So the interesting thing about disaccharides is that based on their biochemistry, some of them test positive and some of them will not. So why does Benedict's solution change from blue to orange? You're not going to be held to the details of this, but essentially for monosaccharides that are reducing sugars, we haven't covered redox reactions yet, but basically what that means is these sugars can transfer electrons to molecules. So in the case of Benedict's, it can transfer it to cupric ions, and when it does that, it'll reduce the cupric ion and basically change the color of it. It'll precipitate and it'll change orange in color. Okay, so a negative result on a Benedict's test would be a situation where whatever you're testing does not change from orange. It just stays blue in color, meaning it does not have monosaccharides inside of it. So when considering experimental design, we want to have a control, and in this case a positive control, one that we know will change to orange, one that we know will test positive in the Benedict solution, is a glucose solution. Recall from class that glucose carbohydrates, they're monosaccharides, they're individual sugar units, and so if we were to make a solution of glucose, that would for sure test positive in the Benedict's test. To do this experiment, we also have to do a simultaneous experiment, that is the iodine test. So remember, Benedict's tests for monosaccharides and some disaccharides. The iodine test tests for polysaccharides. So really in this experiment, the only way that you can know if what you're looking at is monosaccharide versus polysaccharide is you have to subject that mystery carbohydrate solution to both tests. So here's how the iodine test works. You take iodine, which is yellow in color, and you mix it with the mystery carb solution, and when there are polysaccharides in that mystery solution, the color will turn to a dark bluish black. So I've colored it here as black to show you a positive result, result on an iodine test showing that there are polysaccharides. And again, just as an interesting aside, iodine turns bluish black because these long chains of polysaccharides, such as starch, they'll coil up onto themselves, and basically what happens is iodine gets trapped inside those coils and turns black in color. So a negative result on an iodine test showing that there are not polysaccharides inside your solution is that it would just remain yellow in coloration. So an excellent positive control for an iodine test, again, one that we know would contain polysaccharides and thus turn black, would be potato starch. Recall from class that starches are long chains of glucose monosaccharides. So like in other previous virtual labs, I'm going to run through a couple of scenarios that we would have actually performed in class, but now I'm just going to show you the reaction and I'm going to have you record your results in a table and think about the results on a Google Doc for the experiment today. Okay, so the first thing that we're going to test is honey. So we subject it to the Benedict's test. That is, we mix a little bit of honey solution in with some Benedict solution. We boil it for 5 minutes. And lo and behold, for honey, you get a positive result on a Benedict's test. That is, it changes color from blue to orange. We also put honey in through the iodine test to see if it's potentially also somehow a polysaccharide. And the result of the iodine test is negative. So for the iodine test for honey, it does not change. For experiment 2, we soak oats, like oatmeal oats, Quaker oats, in some water for a few minutes, and then we subject that water that the oats were soaked in for a few minutes to a Benedict's test. And your result for the Benedict's test is it's a negative result, so it stays blue. For the iodine test, soaked oats also remains yellow. So record those results in your table, and there will be some questions to think about them down the road. For the third experiment, we're looking at molasses. Molasses is often used in baking. So we put molasses through the Benedict's test. The result of that experiment is that it turns orange, so it's a positive result. We also subject molasses to an iodine test. And the results of the iodine test is that molasses tests negative. For experiment 4, we're going to test sucrose. You may recall from class that sucrose is a disaccharide. So we put sucrose in the Benedict's test. Here is your result for the Benedict's test. We also put sucrose into the iodine test. And here is your result for the iodine test. In experiment 5, we're looking at powdered sugar. So powdered sugar, also known as confectioner's sugar, if you go to a town fair and you put powdered sugar, you put it on, the fried bread or whatever the stuff that comes out of the fryer. So it's confectioner's sugar. It's really just sucrose that's ground into a really fine powder. Okay, so here's your Benedict's result. You also put powdered sugar into the iodine test. And here's your result for that. So you get a positive result on the iodine test. So I wonder what that means. Experiment 6 involves looking at maple syrup. So this is not, the log cabin syrup that has high fructose corn syrup. This is made from maple tree sap, right? So it's maple syrup. Put it through the Benedict's test. You get a positive result in the Benedict's test for maple syrup. We also ran it through the iodine test, and you get a negative result on the iodine test for maple syrup. Experiment 7 is oats again, but here we've cooked them. So we've boiled the oats in water for a certain time, like you would if you were making oatmeal. And the results of the Benedict's test stays blue, so that's a negative result. We put it through the iodine test, and your result is positive, so you get a positive result for the iodine test with oats that have been cooked. Our last experiment is going to look at milk. So you may remember from class, there's lots of different types of disaccharides and monosaccharides and polysaccharides. You're not really being held to know or memorize any specific mono or disaccharide. You will be familiar with glucose by the time this unit is over, and I've asked you to remember a few polysaccharides, but milk has lactose in it. Okay, so the sugar found in milk is lactose. And when we do the Benedict's test on milk, we get a positive result. So milk tests positive in the Benedict's. But when we run milk through the iodine test, it remains yellow. Okay, and we see here, interestingly enough, that it's actually separated from itself. But that's not necessarily an important result. Okay, so that's the data for this particular virtual lab. Again, you're going to have a Google Doc to record these results on, and then I'm going to give you some questions to help you to think about what these results mean. Okay, thanks a lot.